Okay, hi everyone and welcome to BrainMap. This seminar series is co-sponsored by the P41 funded Center for Mesoscale Mapping House in the Martinos Center. It's my great pleasure to introduce our speaker today, Dr. Leonardo Rivera-Rivera. Dr. Rivera-Rivera studied applied physics on his undergrad studies at the University of Puerto Rico, Humacao, and earned his Ph.D. in physics at the University of Wisconsin-Madison for his work on development of MRI method for cerebrovascular hemodynamics assessments, supervised by Oliver Wiben. This was followed by a postdoc in the Department of Medical Physics at the same university, working on accelerated non-Cartesian sampling acquisition and constrained image reconstruction approaches across a range of applications, including motion-encoded pulse sequences and contrast-enhanced MRI. This was supervised by Kevin Johnson. Currently is an associate scientist in the Wisconsin's Alzheimer Disease Research Center at UW-Madison, working in close collaboration with investigators from the Department of Medical Physics, Radiology, Medicine, and Kinesiology. His research interests include development of sensitive markers of structure and function for cerebrovascular health characterization using innovative MRI methods, including data acquisition, image reconstruction, and post-processing strategies. I would just like to remind the audience to please address any question you have using the Q&A box or just raise your virtual hand. Dr. Rivera-Rivera, thank you so much for coming here today. The virtual stage is all yours.
Thank you, Or, for that very kind introduction. And first of all, I would like to thank you and the organizers of the BrainMap series for extending your invitation. I'm very excited to be here, and I hope we have fun while going over this presentation and questions at the end. So just to give a brief overview of the topics that we will be covering today, I will start with an introduction, a sort of motivation into why do we want to do cerebrovascular characterization. Then we'll talk about the basics of flow imaging. And then we will move to applications of flow imaging, largely on Alzheimer's disease and AD biomarker studies, but also in other neurovascular disorders. And then we will also discuss some technical challenges of flow imaging, specifically the patient motion. And finally, we'll go beyond flow imaging and discuss other interesting markers of cerebrovascular health, including tissue perfusions and strain mapping. So on the large scope, the cardiovascular system controls the transport of nutrients and waste throughout the body. But today we'll focus on the cerebrovasculature, including arteries, veins, and the capillary bed. And as many of you know, the brain oxygen demands are quite large, despite of only compromising 2% of the body weight. it consumes up to 20% of the body oxygen supply, which makes it a sensitive organ to vascular disease. For example, even distal measurements of pulse wave velocity, which is a marker of arterial stiffness, are associated with increased cerebrovascular dysfunction. And traditionally, vascular disease is characterized using systemic markers such as blood pressure, cholesterol levels, and genetics. And to probe vascular disease in the brain, patients typically undergo an MRI that assesses structural markers such as white matter hyperintensities, microbleeds, and diffusion. With recent advances in quantitative MRI, functional vascular imaging is also possible, including dynamic perfusion and flow imaging. So it's very important to be aware of the vascular landscape diversity, which includes the macro and micro circulation and tissue dynamics. And it's this diversity that drives the development of tools to characterize the vascular system. For example, the capillary bed can be probed using tissue perfusion techniques, such as contrast-enhanced CT, oxygen-15 PET, and MRI, either with endogenous or exogenous contrast media. To probe the arteries and the veins, typically, these are studied with CT and DSA angiography. However, DSA, even though it offers high spatial and temporal resolution, it exposes the patients to ionizing radiation and is challenging to extract quantitative measures. Another option to probe arteries and veins in the brain is using Doppler ultrasound, and the TCD is inexpensive compared to other imaging modalities, and it offers high temporal resolution, but it also has some limitations, including beam penetration bone window. So alternatively, we can use MRI to probe the arteries and the veins, and more specifically, we can use phase contrast flow imaging. And before discussing flow imaging, let's recall that on an MRI system, there is a net number of spins aligned to a strong magnetic field, and the spin system can be excited by a high-powered radiofrequency wave, and eventually the system will radiate a signal that we can detect with an RF receiver. And a great thing about this received RF signal is that it can be modified after excitation using magnetic field gradients to encode position. And this also makes the scans so loud. So importantly, the signal remembers history in the phase. So if we play these encoding gradients and sample sufficient frequencies, we can then use the Fourier transform as a modalities is the ability to manipulate contrast. So by modifying the pulse sequence parameters, you can generate bright blood, bright fluid, dark fluid images, depending on your clinical needs. And the image phase is often uninteresting because it has limited contrast, unless you're doing susceptibility mapping, which does use this phase. So we can also encode motion with MRI, and on a simple pulse sequence timing diagram, you first excite your spin system, and then you play the gradient that will introduce some phase accumulation. Then you play that gradient again, but with opposite image polarity, opposite polarity, so that now only the moving spins will accrue a net phase. And this will be followed by your imaging module. So from such acquisition, you will get images like this. On the left is the phase image without motion encoding, and on the right with motion encoding. And in the motion encoded image, you can see a vessel from the phase accumulated by the flowing spins. So by taking the difference between these two measurements, we get a phase image that should only be dependent on the spin's velocity. Also, typically with phase contrast imaging, the magnitude images are unaffected. So unless we play really strong gradients, such as those used for diffusion imaging, there shouldn't be large differences between the magnitude data without and with motion encoding. And data collection can be gated to the cardiac cycle and average over many cardiac cycles. And then these images acquired at different time points in the cardiac cycle can be combined, leading to cardiac resolve flow imaging. And in this example, what you're seeing is the magnitude and velocity data of a beating heart, where you can see the thoracic aorta and the aortic valve. So to generate these velocity images, we have to make a few assumptions to the signal model, which are, of course, not always true. And these approximations include that there is no acceleration, there is approximately 1 velocity per voxel, and we can separate the encodes. Then we can relate the phase on the image to the first gradient moments, the velocities, and a background phase term from the coils of resonances, errors in spatial encoding, etc. And commonly, flow imaging is done with a targeted 2D plane oriented orthogonal to the blood vessel of interest. And subsequently, the velocity is integrated over the vessel cross-section to derive flow rates over the cardiac cycle. And 2D phase contrast MRI can be extended into 4D flow imaging with time-resolved 3-dimensional flow encoding. However, MRI is a slow imaging modality, and just acquiring a 2D image can take around a second, and a 3D volume could take over a couple of minutes. And if it's 4D flow, that's 3D volumes plus 20 temporal volumes. It's just unfeasible to do it with some kind of acceleration scheme. In addition, 4D flow imaging of the brain poses unique challenges compared to aortic imaging. For example, brain imaging involves large volumes of interest to cover the vascular tree. And while we don't need respiratory gating, we need high spatial resolution to image the small vessels, contrary to aortic imaging, where we have a tailored geometry of interest and quite large vessels. So traditionally, neurovascular 4D flow MRI clinical implementation has been burdened by these long acquisition times. However, today, there are many different approaches that can be used to allow for clinically feasible scan times. And one way to do it is to accelerate the 4D flow acquisition. And we could exploit the fact that the bulk image contrast information is located at the center of K-space. And using a radial sampling trajectory, we can heavily oversample the center of K-space, but save imaging time by undersampling higher frequencies. And this leads to accurate images with 10 to 30% less data, given that the images are sparse or compressible. So this approach allows us to collect neurovascular 4D flow scans at 3 Tesla with whole brain coverage around 0.7 millimeter spatial isotropic resolution and 50 milliseconds temporal resolutions in 6 minutes. Also, another advantage of using radials is the inherent robustness against motion artifacts due to the short echo times. And the undersampling artifacts are relatively benign. They manifest themselves as streaks mostly. And here you can see an example of velocity streamlines along the circle of Willis. So with flow imaging, we can measure many interesting hemodynamic parameters. And moreover, we can generate these visualizations where we can track cardiac resolve and time average flow patterns along the vasculature. So now that we know how to do fast neurovascular 4D flow, let's switch gears towards the application of these methods to study vascular contributions to dementia. So traditionally, dementia was thought to be driven more or less by separate etiologies. However, more recently, the revised view is more focused on an overlap representation. And in this modern view, cerebrovascular disease has been identified as a major risk factor for dementia due to Alzheimer's disease. AD is the most common cause of dementia, and we don't have any good therapies for it to treat it. Therefore, it's important to understand the interactions between cerebrovascular disease and Alzheimer's disease. And one of the major limitations to study these interactions is the lack of sensitive and specific markers of cerebrovascular health. So we need more specific markers of vascular function to study CBD and AD models, such as the waste clearance lymphatics hypothesis and others that have been proposed. And in this regard, 4D flow MRI is well suited for for comprehensive hemodynamic characterization. And at the Wisconsin Alzheimer's Disease Research Center, we have leveraged 4D flow in AD cohorts. And here are some of our initial findings from a few years back. And in this data, what you're seeing is the blood flow for different vessel segments in the circle of Willis. And overall, we saw a decrease in blood flow for almost all the major vessels in clinically diagnosed Alzheimer's subjects compared to age-matched controls. Those are these red and brown bars. And pulsatility index, the plot to the right, which is a marker of vessel compliance, but was higher in AD compared to age-matched controls, also in multiple vessel segments, suggesting a decrease of vascular compliance in AD subjects. And we also have a few participants with mild cognitive impairment and a younger, relatively healthy cohort, which are these blue and purple bars. And here's another examples of pulsatility index maps derived from flow measures, showing higher pulsatility along the vasculature of AD subjects, followed by MCIs and age-matched cognitively normals. PI is easy to obtain. It's a marker originally conceived as a surrogate of vascular resistance. However, that has been disputed too. So it would be better to have markers with a distinctive physiological meaning. And one of such markers is pulse wave velocity, which is the gold standard non-invasive biomarker for arterial stiffness. And typically higher pulse wave velocities are related to high blood pressure, stiffer arteries, while low pulse wave velocities are more related to elastic arteries. And it's relatively simple to calculate, just the distance traveled by the cardiac pulse pressure over time. And more importantly, carotic to femoral pulse wave velocity measurements has been associated with deposition of amyloid beta in non-demented individuals. And as you know, amyloid beta is a hallmark biomarker and protein of Alzheimer's disease. And pulse wave velocity is typically measured using applanation tonometry, which is easy, inexpensive, and accessible, but it also has inaccuracies. It estimates the distance from the body surface. There are no 3D considerations, and vessels are not always straight. So with 4D flow, we could measure, we could get closer. First of all, we could get closer to the AD biomarkers, closer to the tissue. We could measure potentially intracranial pulse wave velocity, overcoming some of the limitations of the established methods. However, intracranial pulse wave velocity requires high temporal resolution. And one way that we could increase the temporal resolution of the acquisition will be just to reconstruct more time frames. However, that would also increase the noise. So to denoise our images, we could use a constrained reconstruction method, such as a local low-rank representation that have been shown to denoise highly undersampled data sets. And in addition, we could also split the flow encodes during the image reconstruction step to further increase the theoretical temporal resolution of 4D flow. And here's a cartoonish representation of how we can leverage a low-rank reconstruction with flow encode splitting to increase the temporal resolution of flow imaging. On a standard 4D flow recon, we usually need 4 measurements to encode velocities in X, Y, and Z directions and to remove the background phase. So in this example, we are using a 4-point reference velocity encoding scheme. And here we have a cardiac waveform that we want to reconstruct. And we start by dividing the cardiac cycle into multiple bins where data will be shared and averaged. And a timestamp long enough to include all 4 encodes is defined and used to assign projections to bins during the reconstruction. So this enforces that the data with the same projection angle is added to the bins, which also sets the theoretical temporal resolution limit to 40 hours. And after enough data has been collected, we have a relatively uniform sample case space for each of the encodes. Then we do grading and a sense reconstruction, and we obtain images for each of the encodes where the reference image is subtracted from the velocity data to remove the background field offsets. However, if we shorten this timestamp and we split the encode, say for example into 2, the resulting overall coverage of case space for each encode pair will be different. And after reconstruction and image subtraction, the different projection angles on each encode pair will lead to different artifacts, which can end up as residual artifacts in the velocity data. So instead, if we add a low-rank constraint, information from all the bins is used during the reconstruction, and the missing projections are recovered to a certain extent, ultimately helping reduce the potential for residual artifacts. And here is a simulation to help illustrate this better. which combines a local low rank with flow encode split. We have two phantoms with two vessels each, and these phantoms were generated using opposite velocity encodings or VENCs, and were radially sampled using a golden angle distribution and 1,000 projections on a 2D grid. Then the sample raw data was reconstructed using a standard sense and a local low rank constraint, with and without encode split. So after reconstruction, the first encode was multiplied with the complex conjugate of the second to generate the resulting time series. And from this time series, we can see that these local low rank reconstructions are less sensitive to undersampling and residual artifacts compared to the standard sense with and without encode split. And with this approach, we measured intracranial pulse wave velocity in clinically diagnosed AD subjects. And what we found was a statistically higher pulse wave velocity in AD when compared to controls. However, sorry about that. Very interestingly are these two groups to the right. These are healthy middle-aged adults at low and high genetic risk of dementia due to Alzheimer's disease. And the group at high risk, the far right, had parental history of dementia due to AD, and they were also ApoE epsilon 4 positive. And this group of normal subjects was showing higher pulse wave velocity measures compared to those at lower risk. Overall, pulse wave velocity increased with age in the healthy, while it suggests that there is a arterial stiffening happening in AD patients. And Alzheimer's disease appears to accelerate the aging effect. So far, the studies that I have shown you here were limited to clinically diagnosed Alzheimer's disease subjects. However, neuropathological data indicates that there is a misdiagnosis happening over 30% of the time, which is huge. Nowadays, with a few biomarkers that are available to diagnose Alzheimer's disease, including amyloid and tau proteins measured from PET scans or CSF, and more recently, blood plasma markers combined with neurodegeneration from a structural imaging, we have a more biological definition of Alzheimer's disease. And these biomarkers indicate that the preclinical phase of Alzheimer's disease can last over 20 years. And recent research has taken these AD biomarkers a step further and recontextualized them in the terms of time. For example, in this top plot, you can see spaghetti plots of amyloid accumulation from PET scans. And what they show is that once somebody starts to accumulate amyloid, these red dots, the rate of accumulation is fairly constant. So using data-driven strategies from an amyloid positive scan, we can now estimate how long has someone been accumulating amyloid. But there is still a lot of heterogeneity in the symptom expression, as noted by this plot on the bottom right. This is just a measure of cognitive decline, CDR sum of boxes, shown as a function of the amyloid time. And in this regression model, approximately after 21 years of accumulating amyloid, you will develop mild dementia. However, please do notice that there is a large heterogeneity in this curve. There are some people that will never develop clinical symptoms or will develop them much after the 21-year mark. And there is another group which appears to be less resilient and more sensitive, which will decline much, much faster. Therefore, to try to understand what drives this heterogeneity in the symptom expression, we are interested in characterizing cerebrovascular health during the preclinical phase of Alzheimer's disease. And we recently did that in a sample of 136 subjects that were recruited from the Wisconsin Alzheimer's Disease Research Center and the Wisconsin Registry for Alzheimer Prevention Studies. 4 groups were established based on cognitive status, amyloid and amyloid and tau burden. And please notice that 3 out of the 4 groups were cognitively unimpaired. And the controls were age and sex propensity matched to the AD biomarker positive groups. And there was a higher prevalence of ApoE epsilon 4 on those that were AD biomarker positive compared to controls. And in this big table, I'm just showing traditional Cbd markers, including blood flow, pressure, heart rates, white matter hyperintensities, and perivascular spaces. And most of these markers were similar between groups. There was a significant difference in the presence of microhemorrhages between controls and AT biomarker positive cognitively normals, perhaps indicating the presence of cerebral amyloid angiopathy. Some of the flow markers here, total cerebral brain blood flow, was actually independent of AD biomarker status after adjusting for neuron-coupled metabolism by brain volume normalization. And with flow imaging, we typically probe the macrovasculature. However, probing the microcirculation is very important as it's closest to AD pathology in the parenchyma. And one thing that we can measure with flow imaging is the dynamic evolution of the cardiac pulse wave as a surrogate marker of macro and microcirculation. For example, the pumping action of the heart generates a pulse wave, and this pulse wave travels through the intracranial arteries, reaching the capillary bed, and then return to the heart through the venous system. So by characterizing the cardiac pulse wave translation, as it travels from the intracranial arteries, brain parenchyma, and finally veins, we can establish the transcapillary pulse wave delay transit time. And these transit times are indicators of pulse wave transmission, vascular compliance of the macro and microcirculation. And then here's a summary of what we found in these AD biomarkers groups, measure these transit times from the internal carotid artery and basilar artery to the superoccipital sinus. And there was significantly shorter transit times, which are associated with faster pulse wave transmissions in these AD biomarker groups, biomarker positive groups. And the changes were also detected in cognitively normal subjects during the preclinical phase of AD, suggesting the presence of vessel and/or capillary stiffness. With 40-flow MRI, we can also study beat-to-beat blood flow fluctuations. And in this example, you can see blood flow changes in the superoccipital sinus, which is a large intracranial draining vessel. And from these fluctuations, we can derive low-frequency oscillations. And from time, instead of being cardiac result, now it's time result imaging, we can estimate this low-frequency oscillation content from power spectral density analysis in the arteries and veins. And from these measures, we observe a significant reduction of arterial LFO content in AT biomarker positive subjects, as shown here on the left, indicating a reduction perhaps of in arterial vasomotion during preclinical AD. And so this is a typical frequency spectrum of a functional MRI signal. And we have shown that we can measure high frequency cardiac pulsations with 40-flow to derive markers of vessel compliance and stiffness. But we can also characterize low frequency oscillations as a surrogate marker for autoregulation related smooth muscle cell-driven vasomotion. And to summarize these findings in preclinical AD, they indicate vascular alterations are happening during the preclinical phase of Alzheimer's disease. However, longitudinal studies are necessary to elucidate potential interaction pathways. We still need to study if these vascular alterations might help explain the observed heterogeneity in the neuropathologic trajectories of AD. And here's an example of these low frequency oscillations. In this plot where you see our blood flow changes during a fluid reading acquisition and another scan with multiple 20-second breath holds in the intracranial arteries and veins. And time result images were reconstructed, combining that constrained local rank model to a temporal resolution of 3.4 seconds, which allowed the assessment of slow flow fluctuations in contrast to the high frequency cardiac pulsations. Now I'll briefly show you a few examples of other neurological disorders where flow imaging can perhaps help understand better. And in here, this is an example of a brain aneurysm, and velocity is streamlines along the sac. And the researchers are currently investigating how the vorticity and other hemodynamic parameters can aid determine the risk of rupture of these aneurysms. And in this other example, this is a patient with a vein of Galen malformation for which we generated pressure map from the flow data that show pressure differences before and after embolization procedures. And the clinicians are hopeful that this method will serve as a non-invasive biomarker and with future research may prove to be an indicator of treatment success and or disease progression. We can also use flow imaging to track retrograde flow. In this example, is in the dura arteriovenous fistulas. So here the incoming arterial blood flow in the fistula is traveling into the right transverse sinus and the superior sagittal sinus, as seen by this red plot here. And this information is very useful to characterize the severity of the fistula and whether to do nothing or do a clinical intervention. We can also, using probabilistic streamlines and displacement corrections, generate these virtual injections from the flow data, where you can place a seat to track the path of blood. And unlike the DSA and an ASL, the seat location can be chosen retrospectively and placed in a downstream vessel segment, for example. And these virtual injections could assist in vascular lesion characterization prior to and after treatment. However, future studies are needed to really understand the impact on improved pre-procedure planning and patient outcomes. And in this particular example, what we're looking is at an arteriovenous malformation. And virtual injections were used for retrograde flow tracking. So in some of these GIFs, you can see that retrograde filling of the nidus from the blue vein was faster, followed by a slower flow from the green vein, reaching then the feeding arteries. And another application for flow imaging is to estimate cerebrovascular reactivity. In this study, some collaborators from kinesiology calculated CVR measures in healthy and older, young healthy and young older individuals using 4D flow, and where they observed some significant changes, reductions as we age in cerebrovascular reactivity. And on their experimental setup, the subjects in the scanner used an MRI-compatible mask that supplied a mixture of oxygen and different levels of CO2. Then the subjects were scanned with the flow sequence after reaching steady state of endotidal CO2 levels. Now, hopefully I have convinced you that the 4D flow is emerging as a promising method for quantitative neurovascular imaging, as it provides multiple metrics of hemodynamics, including flow rates and patterns, wall forces, and stiffness measures. However, also the parameters derived from 4D flow have a relatively high level of variability. And some of this variability is due to biological sources, blood pressure changes, heart rates, hydration, others. But there is also variability from the technical side of the acquisition, from the high levels of acceleration, noise, and others. And you can appreciate that in this result, where even though there were average mean differences, average group differences, there was quite an overlap between the variance of the groups. And we also have to consider the interplay between technical and biological variation, as head motion appears to be disease dependent. Recently, Mulan Jen showed that Alzheimer's disease patients tend to move more than controls in these fMRI studies. And with these MCI participants somewhere in the middle between these two groups. So it is very important that we reduce the technical variability in 4D flow MRI if we really want to use it in a clinical setting and for truly quantitative measures. And in this study, we aim to improve the robustness and reproducibility of 4D flow by harnessing motion correction. So how did we did it? Well, Previous studies have demonstrated the properties of 3D radial sampling can be used and exploited to aid in motion correction. This was done in T1 weighted and quantitative T1 imaging. And the idea is that you have this randomized 3D radial sampling, you do the time-gated reconstruction and generate high frame rate 3D navigators, which then you can co-register to get motion estimates of translations and rotations, which you can use to correct your K-space or raw data, and then you generate your motion-corrected velocity encodes. However, 4D flow is very slow. It's approximately 7 times slower than your standard T1 weighted sequence. You have to acquire multiple velocity encodings, and you have longer TRs. So if we use traditional reconstruction approaches to generate these navigators, we don't have fidelity to correct for motion anymore. We're talking about navigators generated from approximately 43 projections per frame. So one way to overcome this is to use this approach called extreme MRI, which is a multi-scale low rank regularization approach. It has low memory requirements to generate large volumes, and it provides rapid reconstruction as using a stochastic gradient descent on GPU. So in this study, we combined this reconstruction support to generate high frame rate navigators to correct for motion in 4D flow data. And we tested it on a protocol that had navigators with a spatial resolution of 1.4 millimeters and 1.3 seconds. And we performed some experiments in phantoms. It was just a straight silicon tube that was passing constant flow rates, and we moved the table from side to side to generate different states of motions. Then we tested in volunteers, where the volunteers had a scan where they did not move, and in another scan, they were instructed to move their legs every 30 seconds to induce head motion. And finally, we applied this, we evaluated this approach in clinical exam from ongoing aging studies where the exams, the 4D flow exams did not pass the quality assurance step. And they had artifacts of unknown origin, mostly bad image quality. And here's some examples of the phantom experiments. And what you see here are the translations and rotations that were measured for the different motion stages. And the navigator seems to track motion with high fidelity, and the motion magnitude was variable. This is likely because of the manual displacement setup. And here are some of the images, cross-sectional images from the straight tube, showing the no motion corrected complex difference and velocity data on the right. The motion led to substantial blurring in the data. However, after the motion correction, the distortions were largely reduced for all of the motion frequencies. If we look at the quantitative markers of these phantom studies, on the static scan, we did not see any bias from the motion correction approach. However, on the moving scans, motion correction increased agreement with the static scan. And here's an example of the navigator images using different reconstructions. With this multi-scale low rank, we saw a higher navigator image quality could be achieved with better motion tracking and correction, as seen from the angiogram and the motion estimates. However, sense and direct reading displayed poor image quality and limited motion tracking. And here's some of the motions that we measure on the volunteers during the moving scan. And again, we saw high motion fidelity and stability from the self-navigation. And this volunteer 4, we were wondering what happened. And after digging a bit more, we found that she was properly trained as a dancer, which she decoupled her leg motion with head motion. Some examples of the image corrections that we saw on the volunteers. So we see decreased image blurring and increased vessel conspicuity, as shown by the orange arrows after motion correction and when compared to the static scan. And if we look at the quantitative markers derived from this data, motion correction of the movies can reduce measurement variability, increase similarity to static scan values, and we didn't see any measurable bias added by the approach. And importantly, even in volunteer 4, there were quantitative changes when the artifacts were minimally seen on the images. And on the clinical exams, we had 11 clinical exams that failed QC. And these were the average translation measures on those clinical exams on the order of 1 millimeter. If we look closely to the subjects that moved the most, these are the translations and rotations of those cases. And what we see is there is a high degree of motion variability in clinical data. And sorry, that was a bit fast. And here are those cases, the renal quality of the data without no motion correction. And after the motion correction, we can see reduction in image blurring and increased vessel conspicuity in the internal carotid artery and the middle cerebral artery and M3 segments of the middle cerebral artery. And these are velocity streamlines segmented from this data. And renally, you can see the poor image segmentation that was achieved. But after motion correction, you do see a decrease in image noise and increase vessel segmentation performance. Importantly, the quantitative markers of this derived from these exams, blood flow rates in a few cerebral vessels, they all significantly decrease after the motion correction. Also, similar results were seen for the areas measuring the cerebral arteries, and in various vessels for the pulsatility index marker. So we also noticed that bilateral vessel differences of these markers was reduced after motion correction. So motion correction can reduce variability in neurovascular fluid flow data. It's important to notice that it's an issue, especially in vulnerable populations such as geriatrics and pediatrics, and it reduces variability in the measures significantly. And importantly, even subtle motions can impact the quantitative parameters. And a limitation of this study is that it was single vendor and single protocol. And now quickly, going beyond flow, I would like to show you some interesting MRI-based markers of cerebrovascular health. Now we will move closer to the tissue and brain tissue interactions with the vasculature. And typically these characterizations are done using exogenous media where you inject the contrast agents. These approaches are generally robust and offer high sensitivity, but you have to be invasive and the setup can be quite complex. We could use endogenous media. We won't talk about these approaches today, but such as ASL and others. So after injecting in a contrast, the heart distributes it through the arteries, it reaches the capillary bed, and finally it returns through the veins. And normally we will collect MRI dynamic data after a bolus injection. So you begin with a signal model and obtain relaxometry measures, and this is followed by kinetic modeling to determine perfusion parameters. So a very interesting contrast agent for enhanced MRI that we could use is ferumoxytol, which is an iron supplement, which is used as an iron supplement therapy in patients with chronic kidney disease. It's an ultra small superparamagnetic iron oxide with strong T1 and T2 star shortening effects, and so far has an excellent safety record. However, there are still some concerns over anaphylactic reactions. But one of the main advantages of ferumoxytol over gadolinium-based contrast agents is that it's intravascular, it's unstable with very long plasma half-lives of around 14 hours, which make it suitable for steady-state imaging. And here you see some examples of an MRI acquired at 3 Tesla after injecting 5 milligrams of ferumoxytol per kilogram. We can also try to measure tissue pulsations using cardiac resolve ferumoxytol enhanced T2 star MRI. And from these experiments, before injecting contrast, We did not see any pulsations in gray matter on white matter. However, after the contrast injections, we did manage to measure gray matter and white matter cardiac-induced tissue pulsations, which could be potentially used as a marker for stiffness. There are other modalities that measure brain stiffness. These are more related to wave propagation, such as MRI elastography, and can be used to study extracellular matrix composition, for example. We can try to do cardiac-induced strain mapping using displacement encoded with stimulated echoes. This is an old sequence developed a long time ago for cardiac imaging, strain mapping. However, with advances in data acquisition and reconstruction, we can apply it to the brain these days. And it's similar principles to 4D flow. It's a phase contrast-based sequence. You encode displacements to track tissue motion, and then you can derive from these displacement fields and strain measurements. And here's an example at the level of the lateral ventricles, displacement in different directions. And you can see through the cardiac cycle, mostly there is a high degree of relationship of the motions that we're seeing between the movement of CSF and arterial and venous blood. And we have some preliminary results of these dense strain measures in tissue derived in mild cognitively impaired subjects. And it seems that the tissue strain decreases with worse clinical diagnosis. These are controls versus mild cognitively impaired subjects. So to summarize the major points that we have talked today, hopefully, I hope that I have conveyed that qMRI can provide numerous markers of cerebrovascular health that can be used to probe different scales, including the macro and the microcirculation, brain pulsations, and tissue vascular interactions. And 4D flow has become a powerful tool to study age-related changes in large vessels such as the aorta and in smaller vascular territories of those found in the brain. And by exploiting advances in data collection, reconstruction, and post-processing, we can go beyond traditional and structural markers of cerebrovascular disease to more specific markers of vascular health. And using these markers, we can test hypotheses and study the interactions between vascular disease and Alzheimer's disease. A lot of these models of lymphatics, intramural periarterial drainage have been tested quite successfully in mouse models with very interesting findings, but human studies are currently lacking. So we could leverage these approaches. There is still room for improvements, technical improvements, especially to reduce the variability from motion and other sources, and hopefully to establish some protocol standardization. But regardless, I believe the future is bright for flow imaging, but in general, for quantitative MRI, as these approaches continue to be adopted by large clinical studies. And of course, this work is only possible due to the collaboration of many great people and the funding sources. And with that, I would like to thank you for your attention, and will be very happy to answer your questions.
Thank you so much, Dr. Rivera-Rivera, for this great talk. And we now move to the Q&A phase, as you said. So I'd just like to remind our audience to please address any question you have using it Q&A, chat, or just raise your virtual hand. And while people are typing, I'll just use the opportunity. Oh, just a sec. Yeah, I think we have a question from Bruce. Bruce, if you could please unmute. In a sec, he'll be here. Awesome.
Hey, fantastic talk. Super interesting. I'm curious, do you have any experience in applying these 4D techniques to imaging the extracranial circulation, measures from the external carotid, et cetera, and perhaps how they relate to the intracerebral flow? it certainly is an easier target to be able to measure non-invasively. And while there's some differences in the circulation, I'm curious whether you've been able to make those measurements and what you've seen.
Thank you for the question. And measuring extracranial internal carotid artery is also very interesting. I haven't had a lot of experience with it personally, but we have done experiments where we looked at carotid plaque in the common carotid artery and how after endarterectomy the flow changes at the bifurcation. And also, the idea of if you remove plaque, you might be reducing the amount of microembolism that are happening in the brain and whatnot. I haven't investigated too much of the extracranial vasculature, but it can readily be applied to it, and also perhaps is in some ways easier to do than going for the smaller intracranial vessels. I think it would be interesting to see how some of these markers are behaving in the extracranial vasculature. But yeah, I think it will be a fun topic to study. Unfortunately, I don't have a lot of experience with it.
Fair enough. Although you probably have the data in hand, right? I mean,
yeah.
So just not just a matter of looking at it, but at least you might be able to take an early look at it to see if it's worth pursuing. That's
right, and that's a great suggestion, and thank you for it. Yeah, that most certainly is something that it would be nice to take a closer look. Right? Yeah, the data is there. That's true.
Indeed. Yeah, great talk again. Thanks.
Thank you. Appreciate it.
Great. I think we have another question from Avery. Just a sec. Avery, if you could please unmute.
Do you hear me?
Yes.
Yeah.
Oh, okay. Great talk, Leonardo.
Thank you.
So you mentioned you had some experience with doing ferromoxetol enhanced angiography. And you'd mentioned that you could potentially use the fluctuation you see across the cardiac cycle as a measure of tissue stiffness. I guess the examples you'd shown were from gray matter and white matter, though, where there's also, big differences in just the underlying tissue perfusion. So I was wondering, how would you use this as a measure, or how would you tease apart, differences in tissue stiffness versus just perfusion itself?
Right. And I think I should rephrase it, right? It will be capillary associated stiffness, because we have a voxel that is, we assume, is mostly tissue, but we also have a few cylinders of capillaries crossing it. And then when the rush of blood comes in, the T2 star shortening effects leads to this signal decay, which we see as the tissue as sort of tissue pulsations. But in reality is I think more representative of what's happening on the capillaries. What will be interesting is to try to couple it with other modalities such as MR elastography, where then there's more of a mechanical measure of shear stress and wave propagation, and perhaps then we can sort of disentangle the vascular contributions and more of the tissue biomechanics or interactions with the vasculature.
Mm-hmm.
I don't know if I answered your question, though, but
yeah, it's just challenging to know how much. I mean, it's just due to a change in stiffness versus just to an increase in flow more. I guess because it's just a pure blood volume measurement. Is that why generally the ferromoxetol is just blood volume?
Right. Yes. Okay. And you could do also so these are cardiac resolve, which is what I find interesting part of it. you could also do the time average blood volume measures just from the changes of R2 star and whatnot. And you also can do potentially, so that's the nice thing with ferumoxytol. It's a complex setup, it's invasive, and it has these challenges with potential anaphylactic reactions, although it has a relatively safety record. But then once you get it in, you can do many things. And the other example I was thinking was T1 mapping and potentially water exchange permeability, which is a topic that is of very high interest to try to study the integrity of the blood-brain barrier, which it also becomes possible after an injection from ferumoxytol without having to worry too much about the dynamics of the distribution of the agent.
Yeah, it's really interesting. You have quite the arsenal of cerebrovascular imaging tools at your disposal. I think makes a lot of centers jealous. Great work. Thank you.
It's a lot of great people. Kevin Johnson, Oliver Weaven, myself, and a lot of great people that have come by. So, yes.
Thank you. Perhaps one last question from Bruce Jenkins.
Oh, hi. Yeah, that was a great talk. Thank you very much. Nice, clear slides too. Great visuals. So I was interested. I mean, a lot of things were interesting to me, but in particular, the low frequency oscillations, because they looked like they were a pretty good biomarker, both the aging as well as the ability to potentially use it as a biomarker for AD and MCI. And I was curious about two things. One thing is, can you see these same LFOs, in the peripheral, arteries, say, in the legs, et cetera? And have you ever compared the measurements you get from the MRI with what you might get from ultrasound, for instance?
So the first part of the question, we have to reconstruct a high number of frame rates to be able to get the temporal resolutions that will let us do track these low frequency fluctuations. And the sacrifice that we're currently making to do that is to trade off a spatial resolution. So we do lose access to some of the smaller vasculature of the brain, and we are left with larger arteries, such as the internal carotid artery. So I have not applied it in peripheral vessels, but as long as we're talking about diameters of a couple of millimeters, it should be easily to adopt. And comparisons with, for example, TCD, not with the low frequency oscillations, we haven't had the opportunity to do that yet. But what we have compared is these pulsatility indices with the pulse wave velocity measures and TCD and in healthy aging. And they do show a good level of agreement with, I believe it's an R square of, can't remember exactly, it's 0.5 or 0.6. Yeah, but there is more work to be done there. And also to see if we could correlate these low frequency oscillations to try to disentangle them between what drives them, right? There is respiratory induced, perhaps contributions, but also the idea of these smooth muscle cells contractions and more related to autoregulation. So that's something that's very interesting. The arteries have more smooth muscle cell than the veins, so there should be some differences there too. And then these models of AD, the lymphatics, the iPad, some of them said that the high frequency cardiac pulsations drive the waste clearance or partially do. And others propose that it's the low frequency oscillations which do the heavy lifting. So I think there's many interesting questions that we could try to relate to these measures of low frequency oscillations.
Great, thank you.
Thank you.
Right, so thank you so much again, Dr. Rivera-Rivera, for this awesome talk, and we hope to see you next time in person.
Yeah, that'll be great. Thank you for the opportunity.
Goodbye, everyone.
Thanks again.